So Emma, glad we have coffee today because we're gonna need it. We're gonna talk about death.
Death by coffee.
Emma actually found this very cool story for me to look into about these researchers that were trying to find a better way to preserve brain tissue after death. So these researchers are working on pigs, and they found a way to preserve the pig brain for up to 10 hours after death.
Zombie pigs.
Zombie pigs! Back from the dead.
So everyone was kind of freaking out because they're like, what does this mean for life, death, the zombies? Is it possible?
Exactly, yeah. It could change a lot of things. One of them you mentioned, what do we classify as death? So clinically, death is defined as when you no longer have a pulse or a pupil response. But that says absolutely nothing about your brain activity. So people have seen that you can have brain activity for up to 10 minutes after death.
Wow.
And I mean, we've heard stories of people coming back out, minutes after.
Right. Why do people only have brain activity 10 minutes after death? Because we know the brain's a super complex organ, and it seems like if it could be 10 minutes, why not an hour or 2 hours?
Right. Well, one thing that your brain really needs is oxygen. I mean, most tissues in your body need oxygen. That's why you have blood coursing through your veins, delivering oxygen to all sites. But the brain is especially sensitive to loss of oxygen. So I guess after that 10 minutes, things kind of start to break down.
Yeah,
Especially, it's like an energy-consuming organ.
Yeah, I think it's one of the most, along with heart and
Yeah.
muscle. So that makes sense that if you're not able to produce energy, run out after a while.
Show's over. And I just thought it'd be kind of interesting to mention that the way that we know the activity can last up to 10 minutes after is from this study of patients that have a DNR, so a do not resuscitate. They attach electrodes to them and kind of monitored the activity after they were disconnected from life support. So that's how we know it can last up to 10 minutes. And it's very patient by patient dependent.
That makes sense, depending on if you are more sensitive to oxygen and that your body can carry a little more, then maybe it would promote it longer.
Yeah. And they didn't comment on this, but and it was a small sample size, but I did notice that the men had electrical activity for longer than the women.
I guess that would make sense with their ability to have more muscle and probably be able to uptake oxygen better.
And speaking of electrical activity after death, I mean, you probably have heard stories of people having near-death experiences and seeing, a light at the end of the tunnel, or seeing people in their lives meeting lost relatives. And this can all be explained by brain activity that is still going on. So the tunnel vision that people see is caused by actually a lack of oxygen. If you lose oxygen, you start to see this tunnel vision, so that can explain that. And then the rest of it is just kind of dopamine, which is a neurotransmitter that is misregulated and affecting different areas of the brain associated with memory and emotions.
That makes sense. People have studied brains of people who have the DNRs.
Right.
How do scientists study brain in the lab?
Yeah, that's a good question. So, organ donors, sometimes their organs go to scientific research. So we've been able to take human brains from patients that have passed away and actually extract those cells and culture them in the lab. And just because the body is dead, that doesn't mean that the cells are dead. So if we are able to give them the proper nutrients and oxygen, we can keep growing them in the lab. There are some limitations to this. Two of the biggest ones are when you take those cells out of context, you lose the 3D shape. And what I mean by that is that, cells aren't just alone. They have cells on either side and cells above and below. And it's an intricate network. So when you move that into the lab, we generally culture the cells in one single layer. So you have a cell on either side of you, but not above and below. So that can really change a lot of things in the cell.
Yeah, for sure. And another thing that can change is what's called the mechanical environment of the cell. So in the brain, the cells that are all piled on top of each other, they're not very stiff. They're kind of soft and malleable. If you ever dissected a pig brain in high school or college, you can remember the brain's kind of like a mushy little piece of stuff. And when we do this culturing in the lab, we grow these cells on these plastic dishes, and plastic's really stiff, so it doesn't really respond that well if you're trying to move it around because it's a stiff substance. So that can completely change how the cells are expressing their genes, if certain signals are turned on or off, just because of that mechanical environment. And this is known as the field of mechanotransduction. So if they normally grow these cells in like a single layer on a petri dish, what do they do with these pig brains? Because it sounds like it's a completely different system.
Right. They basically said, why reinvent the wheel? They just left the brain there.
In the pig head.
Yep, in the pig head. Well, I think they did dissect the brain out. So they take the brain out, and then to give it the nutrients that it needed, they used the veins that are already there, and they pumped this synthetic blood through the veins using this contraption that they had built. So this contraption is really sophisticated. It has a pump to pull the blood through the brain. It has an oxygenator to oxygenate the blood. And the actual synthetic blood that they use, it was hemoglobin based. So that's to deliver the oxygen. But it didn't have any cells in it like our blood would have, so it's not exactly like blood. Let's see, they had a computer hooked up to all of this so that they can monitor how quickly the synthetic blood is coursing through the brain, and they were able to adjust the speed, so they picked a physiological speed that kind of like the rate it would be if your heart was pumping, or if the pig heart was pumping. It was also temperature controlled to be at body temperature.
That's good. Yeah, I read that they had like 300 pig heads they did this with, which is just, I can't imagine, because they got these pigs from a slaughterhouse, so they were trying to use what they could.
Yeah.
Which is pretty cool.
Preserving tissue is a big problem in the field. You know, when somebody passes away, you have to immediately preserve that tissue, and sometimes it can take some time to get in touch with the researcher, and the organ can kind of sit around for a couple hours. It's pretty typical.
Yeah.
They call that the postmortem interval, or PMI. And the PMI for samples that I work with, it can be up to like 24 hours.
Wow.
That it sits there.
That's crazy.
And in those 24 hours, like, the cells are dying.
Yeah, and kind of so many expression changes. And just...
Oh, yeah. The cells are freaking out. For this experiment, they decided, okay, let's be realistic and say that it took 4 hours for us to, get the researchers there, get the equipment ready. And after 4 hours, they hooked the brain up to this system, pumping the synthetic blood through. And they maintained it like that for 6 hours, pumping the whole time. So that's 10 hours total. And then, being good scientists, they have a bunch of controls that they do. So one of the controls they did was, okay, what would happen if we just left the brain sitting there for 10 hours, not doing anything, just at room temperature, like this happens. Another control was, let's do the same thing, let it sit for 4 hours, but use what people normally use to clear the brain, which is just like an aqueous solution with some salts. It doesn't have any of the hemoglobin, any of the oxygen, so they called that their control solution, and they did that for 6 hours, just like they did with the experiment. And then the final control is, okay, like best case scenario, we get there an hour after, and you pump the control liquid through the brain. So that's like pretty rare. I don't ever see a PMI of 1 hour.
Yeah.
The PMI of 1 hour is kind of like their positive control. This is the best possible scenario. And the negative control is the sitting 10 hours at room temperature. Using their system, they were able to rescue the cell morphology, or the cell shape. It looked a lot healthier with this synthetic fluid that they're pumping through compared to obviously the 10-hour one.
Right.
Yeah. They also saw that there were more neurons present. They counted the number of neurons in different brain regions and saw that there were more when they used the synthetic blood compared to nothing. And also compared to just that control liquid. They also looked at cell death, and they saw there were fewer cells dying, which is good. And then the coolest part is they measured activity in the brain, and they were able to see that the neurons had electrical activity.
Wow. They did make sure, I think they had an anti-seizure
Yes.
drug to make sure that they would never get to the point of where there's consciousness happening because of just ethically with working with animals. I mean, even though I think they thought it'd be highly unlikely for this brain to have restored consciousness just from this, they still try to take measures to prevent that from happening.
Yeah. They also measured consciousness at the end of that. So while they were able to rescue activity in the individual cells, the consciousness wasn't present because they used an EEG or electroencephalogram to that's the only way we can measure consciousness. We're not very good at doing that. But EEG is kind of the gold standard.
Yeah.
And they saw no brain activity by that. So not conscious, but active.
Yeah. Do you think there's any like implications for the field or also ethics from this study?
Definitely for the field, I think it's going to make us reevaluate how we're preserving tissue, depending on what kind of questions you want to ask. If you want to be looking at activity, this would be really helpful.
Yeah.
Because it's preserving activity in its normal environment. So for that, for sure. And then ethics. Yeah, I mean, what is death? I'm not sure anymore.
I was reading something about this study, and they were quoting Princess Bride and something being half dead versus actually dead. That there's a difference. So they're like, maybe it's the case here too.
Yeah.
Mostly dead, that's what it was.
Some people are kind of worried that people will look at this study and think, oh, you know, if there's hope for a severed big head that has been hooked up to this system for 10 hours and they still have brain activity, like, maybe I shouldn't give up on my loved one that is on life support and not donate organs. So
yeah, that'd be really interesting.
Which, I mean, it will be amazing if we can
Yeah.
save people that we think are past the point of no return, but also we need organ donors.